Chapter 3, Binding and Molecular Recognition. We're going to get some binding done. All right, so molecular recognition is the ability for certain molecules to bind to one another in the face of many alternatives. Remember, when we're talking about your cells in your body, there is typically thousands of molecules that could possibly bind. Most of them have some degree of hydrogen bonding, hydrophobic and hydrophilic interactions happening, right? And so the possible combinations are just insane, right? Which is why you have recognition built into almost every portion of your proteins, DNA, everything, right? Okay. So the structure of the complex is formed have to have a particular three-dimensional shapes. This enables these complexes to have well-defined properties because, again, structure dictates its properties, dictates its function, doesn't it? So myoglobin, this is going to bind and store oxygen for when muscles expend energy, muscles expand, that's true, energy, or convert molecules from food into usable forms. Hemoglobin is going to be the version of that that's found in the blood. So this is going to be the one in the blood, and this will transport the oxygen from the lungs to the tissues. Both myoglobin and hemoglobin both use the same group, which is the heme, to bind to that oxygen. Okay? So there is a receptor, typically a protein, and a potential binding partner. This is called a ligand. This is referred to as L typically. The receptor, which is typically a protein again, but does not have to be, is typically labeled R. And these can come together then to form a complex, which would be RL. So that would be the complex between. Note that this is an equilibrium, and what does that mean about these protein-ligand combinations? They can pop in and out, right? And they're going to do that in solution inside your body regularly, okay? Because all equilibriums are going to be dynamic. So comparing the percentage of RL with a concentration of L will actually reveal how tightly R binds to L, how tightly the ligand binds to the receptor. Okay? And you already knew this. That's the same relationship as an acid-base, isn't it? What is an acid-base? Except for a lone pair taking on a hydrogen, right? So it is a hydrogen acceptor. Okay. So again, this is going to be talking about the binding. It is going to depend on the concentrations of your binding partners. And so there is the percentage bound to the ligand right here. If it goes up really fast and then it maxes out and you hit a steady state because you've maxed out your equilibrium, right? What that means is this is very tightly binding, okay? So it binds extremely well. This is what you see with like strong acids and stuff, right? Where it goes up really fast and then you max it out because everything that can be is pretty much done, okay? Now, does this ever actually reach 100%? No, it can never actually reach 100%, right? It maxes out at some number less than 100%, okay? It approaches 100% forever. Intermediate binding. This would look something like this. And so what you see is you see a nice slow growth. It will approach 100%, but it never actually reaches it, right? And you can add ligand for quite a while, and eventually you will max this out, okay? This is weak binding. They're all on the same scale, so it's very hard to tell this. But weak binding, actually, these all look alike, okay? They're just shortened. So literally, this is just this shifted over, right? If we were to expand this out, this is what this would look like. Okay? So if we were to put our X and Y's, so they just show this part, this would look like this. Just on a much shorter scale. Okay? This one would look like this, just on a much longer scale. Okay? So you can think of it as it just moves downward every time. Does that make sense? Okay. So a very weak binding one, just so we're clear, would take a lot of ligand to max out the receptor. okay, to bind all of the binding sites. At any given concentration of L, this would have a low complex formation. Does that make sense? Okay. Okay, so you're going to recognize almost all of this stuff as like acid-base Henderson-Hasselbalch sort of stuff. Okay? And that's because they're really the same relationships because they're just equilibria. Okay? And so L1 half, which is half the ligand that could be bound, versus 100%, right? So 50% of the ligand is bound. And this percentage of R bound to L is going to be where you have half of the receptor bound to ligand, and half would be then free. And that will show up right here. Let's go ahead and talk about estrogens. This is a steroid hormone. This plays primary roles in the development of female reproductive system and secondary sex characteristics. By the way, would you guys like me to share interesting videos and papers that I find with you? Okay. I tend to think interesting, though, also sometimes means controversial. So, just to be clear, sometimes it's bleach. Did you know microorganisms are actually developing so that they're not being killed by bleach anymore? Yeah, bleach is starting to not work anymore. So estrogen receptors will bind estrogens, of course, as you probably could have figured out. Estradiol binds to the estrogen receptor with an L1 half of approximately 1 nanometer. Nanomolar. Nanomolar, which is 10 to the negative 9th molar, of course. And so here we actually see this is estrone. This right here is estradiol, right there. And these are what the structures look like. Notice the very, very similar structure. See that? Mm-hmm. Sure.
So when it says like the binds to estrogen, that means like whenever you have that molar, that concentration, that means 50% of your estrogen bound.
Exactly. You got it. So, well, 50% of the receptor sites are bound. Okay. So at 1 nanomolar, we would be right here on this axis, the L. We would have 50% of our R's bound, which is the receptors. Yeah. Yeah. Because the receptors will always be the limited number, because we can infinitely add L. Okay? And what's going to happen is eventually the 100% is always going to be where we bind all of the receptors. Think of it like a queue at SeaWorld or, a park. Okay? So like there's a limited number of places where you can queue up and go through. And so when you have a very low L, not all of them are going to be bound, right? When you have a very low number of people. But after a certain while, you get enough people that they're all bound, right? Yeah, and that's going to be this 100% maxed. That making sense? Okay. By the way, again, structure dictates function. Yeah, they look very similar, right? Okay. Okay, so the proteins are going to include binding pockets that are going to be complementary to their ligand shapes. And so the estrogen receptor is right here. Can everybody see that receptor okay? This is then a cutout of this shape right here. These would be the R groups. Does that make sense? So this L, this is the pocket right here. These chains here, this is the estrogen. These chains here are the side groups on this protein. Okay? Okay. Okay, so you guys tell me. I'm tired of talking. You guys. Tell me what's going on with this. Can it be too big? Could your pocket, your receptor site, be too big for your ligand? Oh, yeah, right? In which case you would lose specificity, right? The ability to determine which one you're binding. If it's too big, it wouldn't actually bind it. It'd be too big, right? It'd be like a shoe that's 3 sizes too big, your foot just slips out, right? But on the other hand, could it be too small? Yeah, if a shoe's too small, your foot can't fit, right? Okay. Okay. So what are with these groups here? See how like there's an oxygen here, and then there's like polar groups there, isn't there? And then down here, there's non-polar groups, and what are they associating with? Non-polar, right? Down here, polar groups again. What are they associating with? Polar again, right? Yeah. So your polar and non-polar are going to match, and this polar and non-polar is actually what determines what will bind in there. Okay? So if you match the sizes and you match the hydrogen bonding, typically it will bind. Okay? Those are sort of the two major things. Now, one thing that complicates this is these are not normally without, like, remember the pH dictates if this has a positive or negative charge. You can have ions that would also bind in here. There's many proteins that would not bind without, like, calcium and potassium and that sort of stuff. Okay? Which is why you need those metals in your diet. So again, in this case right here, we have a very narrow binding pocket. This is going to be lined with mostly hydrophobic residues because our structure is mostly hydrophobic. This is going to fit neatly around the estrogen ligands. Remember, that's the ligand, the thing that binds to the receptor. The receptor is going to be a pocket, typically on some sort of protein, though it does not have to be a protein. Okay? This would likely sterically clash with a thicker ligand like testosterone. If you try to shove testosterone in there, that has more thickness to it right here. And so if it tried to actually go in there, it would be repelled. It would be sterically hindered, right? Okay. And it would come off very easily. Okay? No, it's actually hydrophobic because, let me see if I can, this one. So if you look at the structure of this, see how it's almost all hydrophobic? It's so that it interacts well with that. Like if this had a lot of polar groups on it right here, then it wouldn't attach to that hydrophobic region. Okay? Did that make more sense? Okay, so right here we can actually see the difference in different ligands and what features can cause them to be recognized or not recognized. This is also one of the reasons, because of this asymmetry right here, what do you notice about this pocket here? These side chains, because of the amino acids and the directions they're facing, are chiral, aren't they? Yeah. So this has orientation, doesn't it? Okay. And what you can quickly see here is that this right here has strong binding, and the only difference between these two is that the hydrogen and CH3 are on different sides. That's it, right? Okay. This, on the other hand, has an order of 29 times almost worse binding, right? And the only difference being that instead of going into the paper for the CH3 comes out, right? Which means that the CH3 nonpolar association is happening in the opposite direction, and it may have some steric hindrance happening because of the CH3 not being in its pocket, as it were. Okay? So remember that binding is a dynamic process. Whenever we talk about binding, whenever we talk about equilibria, they are dynamic processes, right? What does that mean? Do you ever get binding and then it stays? What has to happen with every equilibrium? It has to go forwards, but it also has to go backwards, exactly. And they have to interchange all the time, right? That's an equilibrium. And so, at equilibrium, the concentrations of all species are going to be constant. Yes, that's true. That's what's equal in an equilibrium, right? But the dissociation and association rates must balance. This means there's going to be an internal, all the time exchange, isn't there? In an equilibrium. Okay. Does anybody know what we call it when there's no exchange in an equilibrium? Isn't it an equilibrium anymore? No, it's frozen, isn't it? It's frozen. It's not equilibrium. So myoglobin is a fairly abundant protein. It represents about 0.5% of weight in human muscles. This represents up to 10% by weight in species like sperm whales, for example. Okay? Myoglobin is going to function to bind and store oxygen. It's going to pick it up from the bloodstream and store it in the muscle for use when it's needed. And then it will release it when the levels drop low enough in the muscle. Okay? And under certain conditions. The fractional saturation, this should actually be gamma. You know, the capital gamma. Looks a bit like a Y, but cursive-ish. Yeah. But you know how slideshows are. Can never get the damn Greek letters to work right. So the fraction of all possible binding sites that contain bound oxygen is this gamma. Okay? This will range from 0, which is all sites are empty, to 1, all sites are filled. I.e. 0, 0%, 1, 100%, right? But we're in a fraction, so it's not a percentage. Okay? The oxygen binding curves are going to plot the fractional saturation versus the concentration of oxygen. Again, they're going to be that same equilibria binding curves that we talked about before, right? Yeah. So the concentration is going to be measured in these cases by partial pressure, because that's the easiest way of determining the oxygen concentration. Okay? Because oxygen is typically a gas, right? And so we'll get the partial pressure, P, of oxygen, O2. This is going to be in units of torr. Torr being the most commonly used pressure units, right? By the way, anybody know why we don't do atmosphere in here? Yeah, we'd almost always be like 1, right? And then we'd have to work in like the second decimal place. Yeah. Torr has more units within it, so it works better here. By the way, does anybody know why the partial pressure here can be used? Do you remember back in Gen Chem, and you learned about the partial pressure over a liquid determines its saturation? It's a direct relationship, okay? So it's a linear relationship. Yeah. And so it's easy to do these things in torr. Okay, so P50, this is half saturation of your binding sites. This is very similar to L50, but in this case we have pressure. So P, pressure, our partial pressures. Again, analogous to the L1/2. In this case though, you know, this actually is funny. I never really thought about this, but this is really kind of a bullshit terminology thing, isn't it? This should be L50 because it's out of 100%. This should be P1/2. Wow. They screwed this up, didn't they? Never thought about it. But anyways, you get it, right? P50, 50% pressure to have your binding sites filled, okay? Okay. This is going to occur typically at about 2 torr, which would be, torr is the same as mmHg, millimeters of mercury, Hg, right? Pressure. Okay, so again, that's for myoglobin. This is human myoglobin here. And you can see the P50 here, 2 torr, and then eventually it reaches its steady state right here, in which case you have basically all the slots bound, right? Any more pressure isn't really going to do much. This right here is the structure of myoglobin. It's a single subunit. Or other way of saying that is the tertiary and quaternary structures are the same. Okay. Anybody know where the heme is? Yeah, it's right there, right? Very good. Okay, so the ability of myoglobin to bind oxygen is going to depend on the presence of your heme molecule. Without the heme, it cannot bind oxygen basically at all. The heme group consists of essential iron atom and a protoporphyrin, 9 in fact. These always use Roman numerals, by the way, to differentiate them from the numbers, as in like it's on the second position or there's 2 of them, that sort of thing, okay? This is the one that was identified 9, which is made up of 4 linked chiral rings. So it looks like a crown, okay? And I'll have a picture later, but essentially think of it as like 4 rings that make a crown that then bind the iron to it, okay? So this iron must be ferrous. Ferrous means a 2+ charge on the iron to form to bind oxygen. If it's any other charge, then it can't bind the oxygen in the heme. Okay? So you can oxidize that iron and get the wrong charge. That is totally possible, and a limited amount of your heme groups will have that. Okay? In fact, that's one of the reasons that the iron oxidizes when exposed to air and doesn't work binding the oxygen anymore. Changes color. So the heme molecule here, this is the heme. Remember that this is actually 3-dimensional. And so what you'll see here is here we have the iron 2+. It's sort of sitting on top of those nitrogens with their lone pairs pointing in towards it and binding it. Okay? Now, to complicate this slightly, this would normally be the cation with the ligand bound to it. So, if you ever go into chemistry, that's the terminology. And then you have another ligand that can bind to this, which is oxygen. Okay? Okay, this is the propionate group, by the way. Everybody knows propionate, okay? 3 carbons with a carboxylic acid that's been diprotonated. It's time? Okay, let me stop here. I will see you all next time, and you have a wonderful day. Thank you for letting me know.